And we are live. Hi everyone, thank you for joining this Google Hangout on air. We're talking about drug discovery and how we make new medicines. Joining me are two fantastic scientists, Allan and Raj. Could you both please introduce yourself?
Hi everyone, I'm Allan. I'm Head of Chemistry at Cancer Research UK Manchester Institute CRUK Discovery Unit.
Hi everyone, my name is Chopra. I head Cancer Therapeutics at the Institute of Cancer Research in London.
Excellent. So this topic is very, it's something people are quite curious about because drugs are the cornerstone in cancer treatment, and much of the progress we've made over the past few decades is attributed to all these new treatments we're discovering. But the reason we wanted to have this hangout was to talk a little bit about what goes on behind the scenes in a lab. How do scientists discover new drugs, and what are these designer drugs that people talk about? What makes a good medicine, and so on. So let's start with a question for Raj. You've had many years experience working in both pharma companies and in publicly funded research. Where does the idea for a new drug start?
So the ideas for a new drug can come from many different avenues. So for example, both in industry but also in academia, we have research labs which are constantly looking for the causes or the drivers of cancer. So new ideas will come from our internal research engine. Okay. It can also come through our partners who aren't exactly involved in drug development, but are involved in the biology of cancer, and therefore they will come up with a new idea, a new potential target, and discuss it with us, and we will have a understanding on the basis of certain principles whether that target is viable for drug development. And finally, we all follow the literature. There is constant data being generated in the journals, in public databases. The Human Genome Project, as you know, is coming up with all sorts of sequencing data of how gene expression is altered in cancers. And again, all of that information, the big data that's coming through, informs us in defining which targets we go for. Of course, we've got to be very clear that the targets we go for are true drivers of cancer, that there's good quality of preclinical research that supports it, that even if someone has done an experiment once or twice, say, in one laboratory, can we reproduce it? And even if it's a good target that drives cancer, can we use our existing chemistry, our antibody approaches to really come up with a drug which can be then absorbed by a patient, for example, or be delivered to a patient in the right manner? And finally, the thing we always have to be clear about is the drug target should really ideally be specific to the cancer and not affect normal cells. Now that is difficult, but that is the way we try and prevent the side effects.
Yeah, and that's very important because that's one of the things that lots of people feel badly about the drugs. So Allan, how is it different in a university setting?
I think, as Raj alluded to, finding good drug discovery targets is quite difficult. One of the things that I think would be within a university environment or within a charity environment like Cancer Research UK is it puts you a couple of years ahead of the game. You start to see research as it's emerging from the laboratories, as it's emerging from the group leaders, not having to wait that 2 or 3 years until it appears in the literature, in the scientific journals. So I think it gives us a little bit of a head start. Now that comes at a slight downside in that there are fewer experiments on that particular drug discovery target, so what we call target validation may not be as robust. So an emerging drug target may fall by the wayside very quickly when we start to do some of those additional experiments to ensure its validity. But I think that immediate access to science as it's emerging gives us the opportunity to start developing the next generations of medicines, perhaps much quicker, albeit with higher risk than is the case within the pharmaceutical industry, where they don't have that immediacy of access.
So you both mentioned the word drug target. So for the benefit of people who don't know what that is, can you explain, what is a drug target? What makes a good drug target?
Sure. So I think the easiest way to think of a drug target goes back to a guy called Emil Fischer in the 1890s, and he rationalized that everything that happens in the body, every every process, there's some form of on switch somewhere. And he rationalized it as being almost like a lock and a key. So that there are lots of locks within the body, and by either locking those locks or unlocking those locks, we can start and stop processes. So what we're trying to do is design a bespoke key that will fit that lock. So, for example, the sorts of molecules we work on are three-dimensional shapes, and they're designed to have an exquisite shape to fit just within that lock. And the idea is that they'll bind to one lock, switch off a particular process, in our instance, switching off the growth of cancer, but will do so in a way that doesn't affect all the other processes that go in with the body. And I think that's what we really mean by a targeted therapy, something that exploits a particular vulnerability or a particular weakness within a tumour cell that isn't present in the rest of the body. So by designing those keys specifically and accurately, then hopefully we can get something that's very efficacious against cancer, but doesn't have all the traditional side effects that everybody associates with cancer treatment: hair loss, diarrhea, vomiting, nausea, all those sorts of things.
So it's actually quite simple when you break it down like that. You've got that 3D shape of a molecule, and you're trying to find something that kind of fits into somewhere and jams it so it doesn't do the job it's supposed to be doing in a cancer cell.
On that level, yes, it sounds beautifully simplistic.
Yeah.
It would be beautiful if it were that simplistic. Obviously, it's a difficult process to engineer that selectivity to make sure that we only target the particular process we're trying to interfere with.
Yeah. So Raj, what are these new drugs like? What sort of different forms do they take? Are they all tablets you take twice a day?
So the historical notion of a drug is either a tablet or an intravenous infusion, which you give through your veins, or a tablet which can be absorbed. And that has served us pretty well. A lot of our chemotherapy drugs are, for example, given as an intravenous infusion or occasionally as a tablet form. But more recently, I think what's been a great revolution is the development of antibodies. So just as Allan described the lock and key mechanism within a cell, there are these three-dimensional structures or proteins sticking out on the cell surface. And sometimes they're very specific to a tumor cell, often they're not, or they express at a higher level in tumor cells compared to normal cells. And you can have an antibody which locks in. Rather than using a chemical to do that, you use an antibody which locks in onto these cell surface molecules and switches them off. And often they are drivers of cancer, and that's one way of dealing with the process. Another way is sometimes these targets are not just found on tumor cells, but these targets are potentially found on immune cells. And what has been a great revolution is that you can use antibodies now to switch on the immune system. Rather than switching off tumors, you actually activate the immune system. And these antibodies are now geared to really make a revolutionary change in therapies, so much so that it is predicted that in the next 10 years, more than 50% of all anti-cancer therapies will be antibody-based. There are also other approaches. It's not just about chemistry, that there are approaches of using cells as therapy. Using one's immune system, boosting it, and targeting it directly against cancer cells using gene therapy approaches is one other concept that's coming through. And then finally, one can use understanding of how DNA and RNA work is to exploit DNA and RNA and use those as therapeutics themselves, manufacture them artificially using chemistry approaches, and then delivering them back to the body to switch off activated or damaged genes in cancer. So we are now moving beyond the classical chemistry approaches that we've all become used to a whole new era of biologics and cell therapies.
So it's interesting you bring up the immune system. We did a Cancer Research UK hangout on immunotherapy last year, and one of the reasons that immunotherapy works is that cancer switches off the immune system, and these new therapies kind of wake it up, as you said. And you can either have antibodies that do that job, or you can almost filter out the immune systems, do things to activate them and put them back in. Correct?
That's right.
Yeah.
So it's what the cancer cell, the immune system is quite an interesting system. On one hand, it helps to get rid of the cancer. On the other hand, it can be hijacked by the cancer to be either switched off, or actually in certain cases use the immune cells to actually help the tumor growth. And so we now are understanding the mechanisms, the two-phase mechanisms. Can we make the immune system anti-cancer and take it away from being a pro-cancer or a driver of cancer? And that is where a lot of effort is being undertaken, not just using antibodies, but I think using the chemistry approaches, the small molecule approaches that we talked about, developing chemical drugs to stimulate the immune system. The thing about antibodies is that whilst they do work, finding the right targets is not as straightforward, but also the manufacturing is very expensive. And using more than one or two antibodies really is, at this moment, possibly not practical. And therefore a combination of a chemical or a drug based on chemistry plus an antibody is the preferred way to move forward.
So you both mentioned another term, target validation. Can you tell, so Allan, can you tell a little bit about what target validation means? And at this stage, if lots of drugs are lost, does that mean the science behind it was wrong?
I think target validation is a long process. In simple terms, target validation means when we discover a new way that we think might be useful for treating cancer, how do we show that it is actually fundamentally involved in the growth and the development of that tumour and isn't just a consequential observation? And that starts right at the beginning of the process. Excuse me. Now, it starts right early on when that biology is starting to emerge, but there's a quote that's often bandied around that says target validation only happens once you have a drug on the market actually treating patients. That's the only time you've got that process absolutely correct. And target validation is very difficult to do. Cancer is incredibly complicated. The human body is incredibly complicated. So to try and dissect that back to a simple biological experiment to say, is particular drug target A involved in cancer, and can we design a small molecule to block that process, is actually a very difficult and complex question to distill down to simple experiments you can do in the lab. So target validation often falls down, and that's not necessarily because the science that it originated from was bad. It's potentially because the science is so complicated and the reagents that one lab has available to them may be slightly different to the reagents that another lab has available to them, so they get slightly different results, which appear to contradict each other. It may also be that they've only done that experiment once or twice, and in the couple of times they've done that experiment, which gave them that initial hypothesis and that initial data, if you do that experiment many more times, then it doesn't look quite as convincing. And I think Raj alluded to this earlier when he talked about the robustness that we need to discover drug discovery. So a drug discovery process from that initial biology idea through to perhaps nominating a candidate that's good enough for clinical development can take 7 or 8 years, and we need experiments that are absolutely robust day in, day out for that time period. So an experiment we do on day 1 can be compared meaningfully to an experiment that we do at year 7 or year 8. And sometimes those target validation experiments in the early stage just aren't robust enough to support that. So I don't think it's necessarily that the biology is bad or the biology is wrong. It's perhaps the biology just isn't robust enough to support drug discovery, or there are subtleties within the ways the experiments were done that give you one conclusion, and if you repeat that experiment doing it through a different methodology, it gives you a different conclusion. Where it's really good is when you get an exciting drug discovery target and you do target validation by 3, 4, 5 different methods, and it all supports the same hypothesis. That then gives you real confidence and real strength that this actually really is something exciting that could potentially be turned into a drug target. And that's really when the excitement of drug discovery starts.
And I guess, you mentioned that sometimes things in those early stages can be quite different when later you try it out on patients. And I guess a good example of that is, we do cell culture in the lab, and that is growing cells in a 2D layer on a petri dish, and you try stuff and it works. But actual whole animals, actual whole humans, organs, then they're 3D. Cells don't grow flat, and it can be very different.
It's a very, very artificial environment, going from that cell growing in a dish, which is supported by an incubator that keeps it at just the right temperature, just the right pH, all the right conditions to grow with all the right nutrients. Actually, cancer cells outside the body can be very difficult to grow, and they can be difficult to nurture. So we have to tailor the conditions to be just right for those cells to support the growth. But in doing so, that makes it a very different environment to the environment that the cancer cell will experience in the lab. And I think the other complexity with trying to treat cancer in animal models or in patients is the cancers that grow there are what we call heterogeneous. They're much more complex. They're not just a single type of cell with exactly the same genetics across that whole tumor. They're much more complicated than that. So that model within the petri dish and the incubator, where all the cells are exactly the same and all respond in exactly the same way to the experiment you're performing, is a very, very different, much less complex case to the more complicated experiments. So things that look very promising in early stage research can sometimes fall down when you start to take account of that intrinsic complexity that we see in patients.
Yeah, and one thing we're increasingly starting to learn is that a tumor isn't just a collection of cancer cells. There's immune cells in there, there's blood cells, there's all sorts of other things going on, and to study that is an immensely complex process.
Yeah, and I think one area that is starting to emerge is the understanding of how the patient's own body can be almost, in the same way it can hijack the immune system to evade being detected by the immune system, it can also hijack the underlying tissue that the tumour's growing on, and then that supports the tumour, it nurtures it, it feeds it, provides all the things it needs for that tumour to grow within the body. And it's very difficult to mimic that within our lab experiments.
So, Raj, once you have a promising lead, what are the challenges that can crop up that, along this sort of drug development pipeline that can make this process complex?
So let me just define what a lead is. So we often will look for, we would screen a library of compounds, for example, a chemical library, which may have, for example, at the ICR, we have a chemical library of about 250,000 drugs. Other companies may have compound libraries of up to 2 million drugs, which you will then screen against your target. You can produce your target as a protein, or you could look for a target within the cellular context, the systems that Allan was talking about, and then see how the chemicals, do they actually directly interact with your so-called protein target. And once you've identified that, that's just the beginning, because now you want to have a pharmaceutical or drug-like properties. The drug has to be absorbed, it's got to be distributed around the body and go to the tumor. Often it's like a foreign substance. When you absorb that, it is metabolized by the liver or the kidney, and so we want to make sure it isn't broken down rapidly. And so all of these things require modification of the original chemistry. Often patients with cancer receive more than one drug, for example, so you don't want that drug interacting with other drugs and causing side effects. And then obviously the other worry is, or the one thing that we have to make sure is that that drug doesn't cause too many side effects, or as is tolerable for the patient. And then, so all of these things are being refined once we define early chemical matter, for example. And that's the same for an antibody. Once you identify an antibody, you want to make it suitable so that you can give it to a patient safely with the right dose and the right ability to target the tumor as opposed to normal cells. And then the point is, if you ever develop a drug, it's got to be different, it's got to add value over and above what the existing therapies are. So, all right, you've got a target, etc., but is it any better? Is it any different? Is it differentiated? Is something that we always think about very early on. And then the final thing, sometimes it's ignored, is that resistance to drugs occurs very rapidly and can occur because of this tumor heterogeneity. You know, the different tumor in tumor, you've got different cells with different chemical pathways switched on, and so if you're going to develop a drug, what proportion of patients have the potential to become resistant? And if they do, can you combine this drug early enough so that you actually add to the therapeutics and the benefit to patients? So all of these things come into play at that time when you think, oh, I've got a hit, I've got an opportunity to start. It's actually the beginning of a long journey.
So going back to something both of you said, Allan, you showed that 3D model of your drug. So when you go through this drug target validation process, Raj, you mentioned, is it soluble? Does it get metabolized into harmful things? Can you then look at that molecule and be like, oh, it's this left-hand corner that's poking out that's doing it? And then do you redesign it by chopping off that bit? And then is it an iterative process?
Leave it to a clever chemist like Allan to think about things like that.
Sometimes you can. You know, if you consider the molecule, you could say sometimes we've got an undesirable feature and it's due to this bit here. That's not always the case. I think in terms of developing that initial lead through to a drug, some things are comparatively straightforward. We know how to make our molecules more potent so they interact, the key fits the lock better. In a lot of cases, we can understand how to design in selectivity so they only bind to the one particular lock. Some things are more difficult, and usually we find that the molecule that makes it all the way is a compromise. It can be very difficult to predict how we would design in solubility, how we would design in the balance of actually solubility and insolubility. If you consider what a molecule has to go through to actually get to the tumor, it has to go in through the mouth ideally, it has to dissolve in the stomach, it then has to pass through the gut, so it has to be water soluble for those parts of the journey, but then it has to be absorbed through the gut, so it has to be sufficiently not water soluble that it can penetrate through the gut. So there's a real balance of properties, and we've got to fine-tune that quite exquisitely to make sure it can pass through into the gut. Then it has to get through the liver. The liver's whole job is to break down things like the molecules we dose to patients. So we have to be aware of how we can design in that stability, and that's not always straightforward. The body almost always throws us up surprises as to, we think we've designed a molecule that's stable. It appears to be stable in the experiments that we can do in the lab. But when it actually is dosed to a living system, parts of the molecule fall off, or it breaks down in new and unexpected ways that we couldn't predict. And then once it's got through that series of challenges, it then gets into the blood. It has to then get as far as the tumor, so it has to last for long enough to get to the tumor, and get to the tumor with enough of it still there to actually block all the locks that are available to ultimately kill the tumor. So there's a whole series of stacked challenges that the poor old molecule has to survive before it can get and actually do its job. Some of those are predictable. We have some really good computational models that can help us predict some of those properties. But some of it's still, because the body is so complex, some of it is still simply trial and error. We make molecules that we think have all the right properties. We do the experiment and we find out that they have a weakness or a vulnerability somewhere in the structure. And ideally, that can be fixed with a small change to the molecule. The chemist can make the next molecule along the line and try and fix those challenges. But sometimes, as with target validation, the chemistry just gets to a point where it just cannot solve those problems, and you have to go right the way back to the start with a new lead compound and start the whole process again. That's incredibly frustrating, but that's the challenge of drug discovery, and that's why it can take 7 or 8 years to go from that initial idea right the way through to something that's available and ready for clinical evaluation.
Yeah, and Raj, you mentioned like drug resistance, and as we know, that's really becoming a problem. We have a treatment that might work for a patient and think, yes, this is amazing, and then unfortunately a few months later you find out that doesn't work. How do you plan for that? And you mentioned that using combinations can kind of delay that process. Can you explain a little bit more about that?
Yes, so one of the big problems with as we said, cancer, even within one individual, is not one genetic lesion or one uh, pathway that has gone wrong or a set of proteins that have gone wrong. It is a number of mutations or taking place. For example, in some tumors, for example, lung cancer, by the time a patient presents with the disease, they may have something like 50 to 200 mutations in a cell. And not all of those are in the same cells. They might be in different cells. And this is so-called tumor evolution. Essentially, you get your initial mutations, but over time, you evolve and your tumor adapts to its environment, gaining further mutations. And in fact, as has already been mentioned, the immune system, but the microenvironment, the surrounding tissue also feeds the changes in the tumor. And therefore, it is a complex a set of cells. And when you come up with a therapy which kills a proportion of these cells and leads to the tumor shrinking, what often happens is that the other cells that do not have that lesion, or the target that was the subject of the particular drug, will actually gain a survival advantage and start growing back. And so, therefore, if you can combine 2 or 3 drugs which take out more than 1 or 2 or 3 lesions, then I think that is a great advantage. However, the problem there is combining more than 1 drug, 2 drugs or 3 drugs is not without its side effects either. So as you add more drugs, you get a side effect profile. And so the other approach that we're taking is to harness the immune system. Actually, paradoxically, the more tumors, the more mutations the cell has got, the more it stimulates the immune system. And so if we can waken up the immune system in the face of lots of mutations, actually that is a real powerful way of overcoming what we call this tumor heterogeneity. The other thing that we're working on is actually to switch on the microenvironment, because that is independent of the number of mutations you get. So if you can reawaken your connective tissue, the cells that are supporting the tumors, then maybe that is another way of overcoming heterogeneity. The third way actually gives us hope, and there's already some evidence for that, is that even though a cell has got 200 mutations, why does it survive? We always think of cancers are really all-powerful. Actually, they're teetering on the edge. They are just about making it because they've got a genome that is totally bastardized. And so they're on the edge. They rely on nutrition in a very different way. They rely on, they respond to the stress created by these mutations in a very different way. So by understanding those pathways and switching on the adaptation that the cancer has, despite all these mutations, is something that will help to overcome the resistance and the heterogeneity. So there's a lot of work around that. In fact, that is the key theme around which we're developing our research program and our ideas and thinking.
I think, sorry, if I can just add another comment to that, I think one of the other areas that is helping tremendously is the research that we've got going on in early diagnosis. Mm-hmm. So there's mounting evidence that this tumor heterogeneity, this complexity, and this almost intrinsic drug resistance actually comes in quite late in tumor evolution, but it happens before the tumor becomes detectable in many cases. Mm-hmm. So if we can get better at early diagnosis and finding those tumors early before that drug resistance sets in, then hopefully treating with 1 or 2 drugs that are perhaps better tolerated may give us a real way of extending the lives of patients with kind of better tolerated and less complex treatment regimes. And I think that that parallel stream of research of early diagnosis alongside the drug discovery that we've been talking about and alongside the immunotherapy, bringing all these approaches together will be a really powerful way of extending the lives of patients and the quality of lives of patients that are living with cancer at the moment.
Yeah, that's a very good point. It's not a one-size-fits-all approach as well. It's almost like you say, cancer evolves and it's a very individual genetic disease, so it's almost taking all these myriad of complicated factors and trying to incorporate that when you design your drug. So very quickly, because we're running short of time, it's a question aimed at both of you really. What makes a good drug? You talked about solubility and things like that, but obviously all of people are different as well. Do drugs vary? Do you have to consider things like ethnicity or diet or lifestyle when you talk about drugs as well? So Allan, if you want to go first.
Yeah, I think for me, I think you're right. It's about tailoring the drug to the patient. So the clinician has a patient in front of them. They're an individual, and it's about making sure you have the right drug for the right patient at the right time for their individual disease. And ethnicity, and we know that certain ethnic groups break down drugs in different ways, so they get either more or less drug treatment depending how they're dosed. It's being aware of those subtleties and all those complexities. But ultimately, what makes a good drug for me is simply one that works, that gets all the way to the clinic and actually helps patients beat cancer.
Raj, do you have anything?
Yeah, I mean, what I'll add to that is exactly what Allan has said at the end. You know, I think we are learning now, you know, how drugs are absorbed by different ethnic minorities, how you know, different people respond, not just, you know, each individual is different. However, despite this talk about personalized medicine or precision medicine, what is most important when we think about new drugs is that it is innovative. We're thinking on novel mechanisms that overcome existing problems and they deal with high unmet need. So there's still some cancers, such as pancreatic cancers, such as esophageal cancers, where there is less opportunity and hope for patients. And so, to me, a great drug will be something that makes a difference to patients with unmet need.
Wonderful, thank you. And just wrapping up, what is the next step? So you've got your drug and you've tested it. How do we get it to patients? Very quickly, what's the next steps in the process?
So once you've, when you say tested it, the key thing, once you've shown that it's hitting the target, that in your preclinical models it is showing anti-cancer effects, and one uses more than one model, as we already discussed. Not every model is different, and you need at least 4 or 5 models to really be sure that this is worth taking into the clinic. Then you've really got to make sure that the drug is safe and ready to give to patients, and that you've defined the dose, and most importantly, the starting dose. That you start at a dose when you go into clinical trials, that you reach the efficacious dose as soon as possible so you can benefit patients as rapidly as possible. And then obviously, these are the safety, starting dose, and all the properties of a drug that the regulatory authorities and ethics committees look for before they sign off and allow us to go into patients.
Wonderful. Thank you so much, both of you, for your time. Do you have any last words to add before we sign off?
No, thank you very much.
Thanks very much, everybody. Thanks for watching.
Yeah, great. Thanks. Bye-bye.
And on the 22nd of June, we have the next part of this Hangout where we'll be talking about the next step of the process where we test these drugs in patients. So stay tuned for that one. Thank you both for your time.
Thank you.
Thank you.